Good evening, ladies and gentlemen. Welcome to the 9th episode of KSOS Online Journal Club. I'm Dr. Smita Narayanan, editor of KJO and professor at the Regional Institute of Ophthalmology, Trivandrum. With me today are Dr. Valsa Stephen, professor again at the Regional Institute of Ophthalmology, Trivandrum, Dr. Dhanya R.S., additional professor at Government TD Medical College, Alappuzha, Dr. Shane Matthew, Senior Vitreoretinal Surgeon at Chaitanya Eye Hospital and Research Institute, Tiruvalla, and Dr. Thomas Cherian, Senior Consultant in Retina and Deputy Superintendent at Little Flower Hospital, Angamali, and Consultant at Kochi Eye Care, Alwa. The article for discussion today is Changing Trends in the Use of Anti-Vascular Endothelial Growth Factor Biosimilars, Anti-Vegf Biosimilars, Insight from the Vitreoretinal Society of India, Biosimilars of Anti-Vegf Survey. This article by Dr. Jay Sheth et al. Appeared in the Indian Journal of Ophthalmology in February 2021. In today's journal club, we will discuss not only the contemporary knowledge about anti-VEGF biosimilars, but also the utility of surveys to glean information about a particular topic. So we are starting the journal club with Dr. Valsa Stephen presenting the salient points of the article. Dr. Valsa.
Thank you, Dr. Smita. Good evening to all and a happy Onam. Today, I'll be presenting the article, Changing Trends in the Use of Anti-Vascular Endothelial Growth Factor Biosimilars, Insights from the Vitreoretinal Society of India Biosimilar of Anti-Vegf Survey. Authored by J.U. Sheth et al. In the Indian Journal of Ophthalmology, February 2021 edition. So coming to the background, biologics are therapeutic proteins derived from living organisms through biotechnology-based processes. Biologics like anti-VEGFs have revolutionized the management of neovascular AMD, diabetic macular edema due to retinal vein occlusions. The commonly available anti-VEGFs are bevacizumab, which is used off-label by ophthalmologists since it is an anti-malignancy drug, and ranibizumab, aflibercept, and brolucizumab, which are approved by the FDA. Repeated injections are however required for these anti-VEGF drugs, resulting in high cumulative drug costs. And this has led to a search for less expensive alternatives like biosimilars. Biosimilars, as the name implies, are molecules which are similar to the biologics. They are produced by reverse engineering techniques in living cell lines and have structure, pharmacodynamics, pharmacokinetics, safety, and efficacy features resembling the approved biologics. A biologic drug usually takes around 10 to 15 years to produce. However, for biosimilars, less research and development is required, and also fewer clinical trials are also necessary to obtain regulatory approval. Hence, these drugs can be produced in 5 to 10 years, and the cost of the biosimilar is considerably less compared to the innovator molecule. Cost is thus a very important factor in our treatment in determining what medical decision we make and what drug we use. In addition, India's market for biologics is very huge. It contributes to around one third of the Asia-Pacific region. So, of the biosimilars, Rasumab was the first biosimilar of Lucentis to be developed by Intas Pharmaceuticals. It was approved by the Drug Controller General of India in 2015 after a phase 3 trial showed its efficacy in 103 eyes with neovascular AMD. However, in the early batches, there were clusters of sterile endophthalmitis cases, which caused the drug, certain batches to be recalled, and Intas later modified the manufacturing process to overcome this problem. While this study was conducted at a time when only Rasumab was a biosimilar available, now in the market, we have newer biosimilars like Ranisurel, Rani-Ice, and Osiva. So coming to the aim of the study, the aim was to present the outcomes of the 2018 and 2020 VRSI biosimilars of anti-VEGF surveys, which were designed to address the physicians' perceptions of and experience with anti-VEGF biosimilars, and to describe the changes in their perception of the biosimilar agents over a 2-year period. Electronic surveys were sent to all members of the Vitreoretinal Society of India in July 2018 and January 2020 to be submitted in 15 days. And the members' perceptions of efficacy, safety, pricing, and the need for more clinical trials before drugs could be approved was sought. Selection of appropriate pricing for Rasumab was a forced choice. So this was a questionnaire which was sent to all the VRSI members regarding the awareness, whether they were satisfied with the efficacy of the biosimilars, the safety of the biosimilars, whether they thought more clinical trials were required before the drugs could be could be approved, and regarding the costing of the biosimilars. So now coming to the results of in the 2018 study, of the 700 VRSI members, 112 responded, and in 2020, 98 members out of 826 responded to the survey. Of these, majority were aware of the biosimilars, and around 90% felt that approval of biosimilar drugs required larger and better designed clinical trials. Coming to the choice of the anti-VEGF biosimilars, there was an increase in the use of the ranibizumab biosimilar from 2018 to 2020, from 41 to 56%, with a significant decline in the use of bevacizumab biosimilar. The proportion of respondents who were satisfied with the safety and efficacy of ranibizumab biosimilar increased substantially, with a reduction in, but there was a reduction in satisfaction levels of the safety of bevacizumab biosimilar. However, satisfaction with its efficacy remained stable in the survey of both years. Coming to the economics, a substantial proportion, 37% in 2018 and 40% in 2020, considered that the current cost of ranibizumab biosimilar was sufficiently low for it to be used as a substitute for Avastin. But when asked to estimate the price of ranibizumab biosimilar that would convince them to switch from Avastin, it was seen that more were willing to accept a higher ranibizumab biosimilar price in 2020 compared to 2018. So this was the only survey data on physicians' perceptions of anti-VEGF biosimilars in India. However, the study had certain limitations. The participation rate was very low, which limited the interpretation of the results. Also, there was no data regarding the number of respondents in the 2020 survey who were part of the 2018 survey cohort. So we could get only an idea about the overall trend in biosimilar usage among the VRSI members, rather than a direct comparison between the same cohorts. So to conclude, the introduction of biosimilars has given Indian retinal specialists additional anti-VEGF drug options. Retinal physicians are well aware of biosimilars, and there is an increasing trend towards prescribing a ranibizumab biosimilar. Physicians appear to be generally satisfied with the safety and efficacy of biosimilars, but believe that more rigorous trials should be conducted prior to regulatory approval. A third of the respondents felt that the biosimilar is appropriately priced, but acknowledged that a further price reduction would be necessary for it to become the drug of first choice. With patents for biologics about to expire and acceptance of biosimilars growing, a shift from the branded biologics may move towards the biosimilars. Thank you.
Thank you, Dr. Valsa. That was a very concise summary, and also you have told us about the trends very nicely. I now request Dr. Dhanya to come up with the statistics and also tell us something about the survey.
Okay, thank you, madam. Okay, is my screen visible, madam?
Yes, Dhanya, it is visible. Perfect.
Okay. Okay, so good evening, everybody. We'll move on to the statistical analysis and critical appraisal of the study. Okay, so the title of the study is the Changing Trends in the Use of Anti-Vegf Biosimilars, Insights from the White Survey by J. Seth et al., published in IJO, January 2021 edition. So in the abstract, they say that the aim of the study was to present the outcomes of the 2018 and 2020 White surveys. But actually, the purpose or the objective of the study is to analyze the changing trends of the usage of anti-VEGF biosimilars. And the abstract also gives a summary of the methods, the results, as well as the conclusion, and an important conclusion regarding the physician perception of safety and efficacy has not been included. Now, coming to the introduction, the topic is very well presented to the reader. Background information regarding the biologics and biosimilars is explained well. And it also emphasized the need of the study, that is India being an enormous market for biologics, the third highest in the Asia-Pacific region. It also says that the cumulative cost is an important determinant of the medical decision-making and drug selection in most countries, which make a shift towards a less expensive biosimilar. And this is where this study becomes relevant. So the objectives of the study is to analyze the changing trends in the use of anti-VEGF biosimilars among the members of VRSI, and also to see their perception regarding the safety, efficacy, pricing, and need for enhanced clinical trials before regulatory approval of these drugs. Now, the study design, it was an online survey of the members of VRSI, which was conducted in 2 points, in July 2018 and January 2021. So it is a descriptive study. Now, the advantages of this online survey is that they are cost-effective, time-saving. It can collect both qualitative and quantitative data, enables access to large and geographically distributed populations, and the results are also very similar to paper-based survey results. But many people may not consider them as significant, and hence sometimes the actual purpose of these surveys may not be served. Now, the study setting, it is all over India. All members of VRSI have been included. But from this study, we cannot make out if the regional differences is affecting the result of the study. The study population, that is the inclusion criteria, the survey was open for both the regular and the associate life members of VRSI, but exclusion criteria has not been mentioned. Coming to the sample size was not calculated. Hence, we are not sure if the minimum sample size for the survey has been attained or not. Moreover, there was a very low response rate, that is only 16% in 2018 and 12% in 2020. So we do not know whether the study could reflect what it was intended to do, and also it would affect the generalizability of the study. Now, how was this done? Electronic surveys as emails were sent to the members of VRSI in July 2018 and January 2021, and they were asked to complete the survey in 15 days. And this is how the message was given. And when we click here, you go to a Google form which asks your email ID and state and city of practice, and then we'll go to the questionnaire. So the study tool used here was a questionnaire, which was kept as an annexer in the study. It is a user-friendly design, and it has a user-friendly design and layout. That well-designed questionnaires always improve the reliability and validity of the collected data. The questions were short, easy. The answering instructions were very clear, but we cannot make out, or we do not know whether multiple responses from the same device was avoided. And also no questions regarding the general characteristics of the study population, like the age, gender, years of practice, etc., were not included in the question. One of the questions that is regarding the current cost of Rasumab, it was asked as a first choice question. What is a first choice question or response? They make survey respondents to choose a response option that indicates a definitive opinion. So we do not have options like don't, no, neutral responses are not there. So, and usually these first response choices are mandatory questions. Now, what are the study variables assessed? That is, they are the participants' perception of efficacy, safety, pricing, and the need for more clinical trials before the drug could be approved. And all these are qualitative variables. Coming to the data entry and analysis, they say that the data is given as descriptive statistics, but they also say that it was given as frequency tables and donut charts, but these are not seen in the study. And most of the responses are reported as nominal data, whereas the question on appropriate pricing was reported as ordinal data. And data was analyzed using Microsoft Excel as well as Stata 16.1. And comparison of the 2018 and 2020 data were performed using chi-square test, and variables with a p-value less than 0.05 was considered as statistically significant. Now coming to the result, the comparison of the two surveys. Now, first point was the awareness of biosimilars, and it was seen that the awareness of anti-Vegf biosimilars increased from 96% to 100% from 2018 to 2020. But here the p-value is not significant. The reason could be that one of the values in the data is 0. So that could be one point why the p-value is not significant. Even though the result is not statistically significant, it is a clinically significant result. Need for stringent clinical trials. So percentage of respondents who wanted the approval of biosimilar to be made more stringent, that increased from 89 to 91%. Still, here also the value was not statistically significant, but it's a clinically significant result. Now, choice of anti-Vegf biosimilar in 2018 versus 2020. It says that usage of ranibizumab biosimilar has increased from 41% to 56% with a p-value of 0.2, and the usage of bevacizumab biosimilar has decreased. And the value of the ranibizumab biosimilar is not statistically significant, but still this is also clinically significant. Now, the ninth question regarding if you have used biosimilars, are you likely to continue using it in the future? And the options given are yes, no, and not applicable. So this is regarding continuing usage of the biosimilars in future. Here there is a small confusion. That is, they say that the participants are more likely to use ranibizumab biosimilar, which has increased from 73 to 82% compared to bevacizumab biosimilar. Whereas the options are only whether you, if you are used a biosimilar and would you continue to use. So the yes respondents is the 73 and 82, that is in 2018 and 2020. Here, actually, we did not compare the two biosimilars. So this result cannot be said from the study. Now, another graph is given as safety and efficacy of anti-Vegf biosimilars in the two surveys. But it is actually not the safety, it is the participants' perception regarding safety and efficacy of the anti-Vegf biosimilars. And it says that the ranibizumab biosimilar, both the perception regarding safety and efficacy has improved from 2018 to 2020, whereas for bevacizumab, the safety has decreased. The perception of safety has decreased from 30 to 25%, but its efficacy, the perception regarding its efficacy has not changed. These results are also not statistically significant, but clinically significant. Now, coming to the economics of anti-Vegf biosimilars, the price of anti-Vegf injections in India, it is given as a graph. The cost of aflibercept, ranibizumab, as well as the ranibizumab biosimilar is compared. And majority of the respondents in both 2018 and 2020 said that, or they believe that biosimilars have made anti-Vegf treatment more affordable to the patients. And when they were asked to estimate the price of ranibizumab biosimilar, that would help them to, or convince them to switch from Avastin, most of them chose a rate that is $54, which was far less than the actual price of the racimab, and also the willingness to pay price of racimab at which the respondents would switch from Avastin, it increased from 2018 to 2020. That means in 2020, they were ready to pay a higher price for the racimab biosimilar. Now, this question, is the present cost of the racimab biosimilar appropriate if we want to substitute Avastin for? The responses were yes, no, and yes, no. That is, racimab should be priced lower than 130, and the third one was it should be priced higher than 130. Here they say the result that they give is similar proportions of respondents in 2018 and 2020 are of the opinion that the current price warrant a switch from Avastin, but it is only 37% or around 40% have that opinion. Majority says a no, and from the graph we cannot make out whether they want a higher price or lower price than the present cost. Now, coming to the discussion, the study says that there's a significant increase in the use of ranibizumab biosimilar, whereas the use of bevacizumab biosimilar has decreased. The question regarding future use of biosimilars, they anticipate using more of ranibizumab biosimilar, but not bevacizumab biosimilar, but no such data could be concluded from the questionnaire. The validation of biosimilars for unrestricted clinical use requires additional studies that majority of the participants agree with it. And they also say that better designed clinical trials should be performed before biosimilars are approved. In 2020, physicians became more satisfied with the safety and efficacy of ranibizumab from 2018, and they were less satisfied with the efficacy, safety of bevacizumab biosimilar. And the study says that despite the episodes of racimab-related sterile endophthalmitis, there is a trend among the retina specialists in India to increasingly use this drug in their practice. But no questions regarding awareness of the complications of biosimilar among the participants were given in the questionnaire. This study represents the only survey data on physicians' perception of anti-Vegf biosimilars in India. Hence, a comparison is not possible, and we do not know if the values obtained in the survey are similar. And it says that a 2-year interval between the surveys validates the original survey data and identifies trends. And we also do not have the data regarding the number of respondents in the 2020 survey who were also part of the 2018 survey cohort. Hence, we cannot comment on the generalizability and extended validity of the study. The conclusions given are: physicians are well aware of biosimilars. There's an increasing trend towards prescribing a ranibizumab biosimilar, and physicians are generally satisfied with the safety and efficacy of the biosimilars, but they want more rigorous trials to be conducted prior to regulatory approval. And they also say a further price reduction would be necessary for racimab to become the drug of first choice. And there was no financial support and sponsorship, and there was no conflicts of interest. My few suggestions are: from the analysis, a clarity regarding the objective in the abstract has to be included. There should have been some methods to improve the response rate, and general characteristics of the study group should be included, and the two groups should be made more comparable. And here there is no clear data to support the anticipation to use more of ranibizumab biosimilar. And it is a perception of the participants regarding the safety and efficacy of racimab biosimilar that is studied, and not the safety and efficacy of the drug per se. The patent of the parent drug has been over, and biosimilars help to reduce the cost of anti-Vegf treatment and reduce the financial burden to the patient. So the topic is clinically significant, and that is the importance of this study. But stringent studies are required to confirm the safety and efficacy of the newer drugs before they can be approved. So not to compromise the safety. And this is another study I just wanted to refer to. It is published in the British Journal of Ophthalmology, comparing a ranibizumab biosimilar SB11 to ranibizumab in neovascular AMD. It was by the Pressler et al. published in 2021. It was a multi-center randomized double-masked parallel group phase 3 equivalent study between March 2018 and December 2019. And a 52-week analysis of the phase 3 study shows that the biosimilar and the reference ranibizumab have comparable efficacy, safety, and immunogenicity, and the pharmacokinetic profiles up to week 52. And this is another study published in the Retinal Physician comparing the regarding the biosimilars. So we have the parent drug, but many new drugs are coming into the market. Thank you.
Thank you very much, Dhanya. That was presentation that was an excellent critical appraisal. And I think it was very informative to all of those who were listening and all of those who would be listening in YouTube also. Very informative. And I particularly, yeah, you've stopped sharing.
Yeah.
And I particularly liked how you told that the language in studies is very manipulative, like the objective and the conclusion sometimes do not match. And I found it very useful that how if people are learning how to do critical appraisal, they would really appreciate this critical appraisal of a survey. As you have pointed out, this topic is clinically significant. And so today we have a panel with Dr. Shane Matthew and Dr. Thomas Cherian. And I request Dr. Dhanya to conduct the discussion about the anti-VEGF biosimilars. Dr. Dhanya again.
Yeah, thank you, madam. So the importance of biosimilars in the present retinal world, in the retinal world, in the ophthalmology world, it's very important, especially because of the huge economic burden to the patients because of the anti-VEGF treatment, the need for multiple injections, all those. So what are this? So where are the biosimilars taking us to? So we will here today we have two senior consultants in the field of vitreoretina, Dr. Shane Matthew and Dr. Thomas Cherian, and we'll have few questions. So I'll start with Dr. Shane Matthew. So, sir, so what are these biosimilars and how do they differ from the parent drug in their chemical form and the mechanism of action at the cellular level?
First of all, thank you very much for adding me for this discussion. So a few points which I wanted to convey was one thing is obviously a survey will have its own disadvantages. That is quite normal to have a disadvantage. But when you look at it, I think the authors did not have a choice but to go for a survey like this to get an abrupt result on the biosimilar use or efficacy pattern. I think that was the best possible way, right? I think that is why they went for the survey is what I feel. And also, I just wanted to ask you was there any mention on why bevacizumab, the trend of using bevacizumab came down? Was there any reasoning? Has the author mentioned any reasoning? I did not find any. Did you?
No, sir. Yeah, it was just given as a result that the use of bevacizumab biosimilar came down.
Yeah. So, and coming to your question, according to WHO, biosimilars are defined as biotechnological products. So basically, they are biological products. They are proteins, and they are comparable with an already approved reference product in quality, non-clinical, and clinical evaluation. So a biosimilar molecule should represent or demonstrate similarities in pharmacokinetics, pharmacodynamics, safety, and efficacy to the innovator biologics. So we have, as readers, we have an immediate thought process in our mind thinking that, is it a copy of the biology or the innovator molecule? It is actually not a true copy of the innovator molecule. There are few subtle differences in the structure, but definitely it is a complex structure, probably as complex as the innovator molecule. So when you think about it that way, the cost involved in the research and development, R&D definitely is huge for biosimilar also. But there are certain other things which makes it cheaper. I think we will come that in the later course of our talk. So now coming to the second part of your question, yes, the chemical characteristics are more or less the same with slight difference. And the mechanism of action is also the same. That is why they are called biosimilar. So I think the word itself explains the real meaning of it. It is a biosimilar.
So one more question to you, sir. What are the types of biosimilars that are available in our market now?
Yeah, so there is an interesting article which was published in 2020. At that point of time, they had almost 28 simulators available in the market at that point of time. And interestingly, soon after the study was launched, in 4 months' time, I think 16 of them got out of vogue because they were not meeting the equivalency standard with the innovative biologics, and a couple of them had mergers. So actually, it is beyond the scope of this talk to enumerate how many biosimilars are available in the market right now, because there are plenty. But I think probably it's pertinent to talk about few of them available in India. So coming to that, obviously, if somebody has to make a biosimilar, the parent drug has to have their patency date extended, or rather the patency time has to expire. Only then they can make a biosimilar. So ranibizumab patency has been expired, and Eylea is almost expired. So in our market, Eylea does not have a biosimilar now, but I think Shilpa Medicare is coming out with one soon. So probably that is what we should be looking forward to. DGCI has given the approval for conducting a trial. Now coming to bevacizumab, we have many. I think Zyveb from Cadila, Bivataz, Bivasural. There are a lot more, but I think less frequently used. I do not have any personal experience in using biosimilars in bevacizumab. And as the previous speakers has rightly mentioned, ranibizumab have a lot of biosimilars right now available in the market, namely Oziva, Rani-Eyes, Razumab, Ranizural, like that. So now coming to adalimumab. Adalimumab is something which is used in uveitis. So that has got a biosimilar coming up. Cadila, I think, coming out with the Cadalimab, which is going to soon come out into the market. And infliximab is yet another drug, again used in uveitis. It will have a biosimilar in the name of Renflexis from the Merck laboratory. So I think these are the few of the ones in the pipeline, which is going to come into India soon.
Okay, sir, thank you.
Yeah, Thomas?
Yeah, as Dr. Shane rightly said, biosimilars are not photocopies of the original drugs. They are in fact done, manufactured by a reverse engineering process. Let me take that reverse engineering in a layman's term. You have a high-end machine, maybe a car or an aeroplane or a computer, something which it's difficult for you to develop in your own place or in your own country and come out with results comparable to the best in the market. So you buy one machine, then undo the machine, make it piece by piece, and study all about the machine and make a new one. That is something similar to a reverse engineering process, how a biosimilar has been made. So it's not an actual photocopy. The drug has been dissected, reverse from the product which is available in the market to the raw material, and then brought back as a new drug, which is a biosimilar. So it's almost similar, but not 100% similar. That is one perception of the biosimilar, which I would like to convey in a layman's term. Then about the bevacizumab biosimilars not being used. The bevacizumab is not a drug which is expected to be used in ophthalmic practice. Nevertheless, we have been using it for financial reasons when we didn't have another option. We didn't have biosimilars. The innovator drugs were prohibitively expensive. So we had to use bevacizumab, but it's a drug used for some other condition in medical science. And the biosimilars are also expected to be used in those conditions, not primarily in treatment for retina diseases as intravitreal injections. So that is why we don't have enough experience with the bevacizumab biosimilars. The reason why we went for biosimilars is because bevacizumab itself was considered an off-label drug. Then came the Avastin Crisis a couple of years back. There was a ban which came in. Then all the doctors' organizations, the All India Ophthalmological Society, the Vitreoretina Society of India, all represented, and the ban was reversed. But at the same time, these brought up many questions whether we should take up all this trouble. Is there an alternative? And that was the time when biosimilars were slowly penetrating the market. So the use of bevacizumab itself came down after that Avastin crisis, the ban and the reversal. So nobody, in fact, or at least hardly anyone, tried a bevacizumab biosimilar in ophthalmology. So that is why we have extensive experience with ranibizumab biosimilars, but not with bevacizumab biosimilars.
Thank you, sir. Yeah, let me move on to my third question, and I ask Thomas, sir, which was the first biosimilar molecule in India, and how it got its approval?
The first biosimilar molecule, the Anti-Vegf biosimilar molecule was concerned. It was Rasumab of Intas, which got the first approval in India. This was a biosimilar of ranibizumab or Lucentis or Accentrix, as we know, the innovator Novartis product. This was done with a phase 3 trial with 103 patients. And the big question was, is 103 a sufficient number to get an approval for a biosimilar? Because we have all gone through the process of getting approval for an innovator molecule. We have gone through the trials of ranibizumab, Accentrix and Lucentis. Then we have gone through the trials of Eylea. Many of us have been partners for or investigators for those trials. And it was an extensive process. Some were involved in phase 1, some in phase 2, some in phase 3. While biosimilar, what we see at it, it got a very quick approval with just 103 patients. But then there are two bodies approving these drugs in India. One is the DCGI and the other is Central Drug Standard Control Organization, or CDSCO. Both these are government agencies under the government of India. And for getting an approval of any biosimilar, not just an anti-VEGF drug, the CDSCO and the DCGI have a number which is much less than the number which is required for approval of an innovator product. So this 103 was not something which the manufacturer proposed. This was the number which the DCGI and the CDSCO gave to the manufacturer to conduct a phase 3 trial and compare the safety and efficacy of this product vis-a-vis the innovator drug. And once that was satisfied, the drug was approved by the DCGI. That's it. So it's not that it was a shortcut or something which was irrational. It was the proper study as per the rules, as per the law of the land, and that is how it got approval.
Yeah, even I think the US FDA also, if as far as the sanctions of biosimilars are concerned, throughout the world, they carry out the same approach. They don't want that many numbers as the innovator molecules had. The reason what they propose is because this is a biosimilar. So we know that it is similar to the parent, the innovator molecule. So all we have to show them is an equivalency trial, a trial to show that it is equivalent to the innovator molecule. So that has to be tackled with a much lesser number. So probably that is the reason. I'm sure we would have gone through, or our agencies also would have talked to the agencies abroad, or would have come to a consensus, and probably that's how this number was taken.
Okay, so yeah. To Dr. Shane Matthew, so this is partly answered already, but I'll just put it once again. The parent drug is very high, like the price is very high, and our biosimilars are around very low, like 50% or less. And now it is available. But how this can be produced and distributed at this very low cost?
Yeah,
How
that's a.
this huge
I
difference
think
is
that
there?
is
Yeah.
that is see.
It is unbelievable to the patients actually when you
Exactly. That is
discuss this to the
exactly
patient.
that's exactly
And
what I
they,
wanted to point out, because many patients ask
yes,
this question back. See, how come this drug is so less expensive? I'm sure the quality of this drug is less. So I think
yes,
as the clinicians, we should be knowing this answer very well, because we will have to use this answer to many of our patients. So here is what it goes. So if you that is why I told you in the initial part of my talk that don't think that this drug is produced just without any R&D. Yes, it goes through definitely a lot of research, and it is also a complex molecules, as complex as the innovator molecule. So the expense on making that molecule is there, but there is a lot of saving done on the clinical trials. So you don't need, such as Dr. Thomas rightly pointed out, you don't need a large clinical trial to substantiate or get an approval. So I'm sure, I think that is the place where they save money, and that is being transferred onto the cost of the drug. I think that is where they are pricing it low. Yeah.
yeah. Okay.
And another point here, the innovator molecule has already, the innovators, the developers, the pharmaceuticals which are producing the drug, they already made their money. They can very well cut down on the cost and bring it comparable to the biosimilars at this point of time, which they are not doing. For biosimilar, the research cost, as Dr. Shane pointed out, is much less compared to the innovator molecule. And they are producing the drug and they are selling it at a lower cost, while the innovators, instead of bringing it down to that level, they are still maintaining that high cost.
Yes, yes, sir. Thomas, sir, is there any difference in the dosing schedules or like for the biosimilars? Is there any difference from the parent drug when you are planning to give it to a patient?
Absolutely not. When I am planning a biosimilar of ranibizumab, maybe be it Razumab or Rani-Ice or Rosiva or Ranisural, and when I am comparing it with the drug Accentrix which is available, the dosage is the same. The only difference comes is when you offer the patient the choice of a newer generation anti-VEGF, be it aflibercept or a brolucizumab. And when the question of affordability comes, the patient chooses ranibizumab or Razumab, the biosimilar Razumab, because you have to offer all these choices to our patients, especially if it is a case of wet AMD or a PCV, where aflibercept or brolucizumab is known to act better. So if that is not affordable to the patient and we go for a Razumab, then the dosage maybe changes. Otherwise, when you are doing a head-to-head comparison with Razumab versus Accentrix, the dosage is absolutely the same.
Okay. Okay, yeah. And one more question to you, sir. Our experience with the parent drug is actually very long, and we are very well satisfied with the safety, efficacy, everything. And how do you think these biosimilars are going to make a huge difference, or do you think that already they are on the top of the list?
Yes, biosimilars have already made that difference. Because when the biosimilars came into the market first, all of us were skeptical about using the drug. We all held on for some time. Not for the first 3 or 4 years, we didn't use it regularly. But then we slowly started using it in the hope that it is going to substitute the off-label drug, which we were using for a long time. Anyway, the off-label drug was off-label and the biosimilar was approved by the DCGI. So we were legally safer when using a biosimilar than the off-label drug. So that is how we started. And we never expected this to substitute or replace the established innovator drug, which was there in the market. But as we went along, we found that the efficacy, there are in fact 3 points which are to be considered while selecting your anti-VEGF drug. First is, of course, the safety of the drug. Second is the efficacy of the drug. And third is the economic part. When we use more and more biosimilars, of course, all our experience is with Rasamab. The other 3 products which are in the market, or maybe the 4 products which are in the market, came in quite late. So all our experience is with Rasamab. As we use more and more Rasamab, replacing the Avastin which we were using, we gain more confidence. Of course, there are a few batches of the drugs giving sterile endophthalmitis, which were managed. We all went through the reports. But then the company itself was proactive in that when the batch 52 had a problem, they immediately stopped distributing the drug. They took back the drug and gave a notice without anyone pointing out and taking them to task. The company itself stopped it. That gave us more and more confidence in using the product. And I don't think any one of us sitting here, or for that matter, anyone in this part of the country, had any complication while using a biosimilar. So after it replaced Avastin, then we gained more confidence. And now we started offering to the patient a drug which is either imported or a drug which is made in India. And that message went back to the patient. Instead of explaining the whole thing, what is a biosimilar, how is it produced, then we slowly started telling the patient that one is imported and one is made in India. And the patients started taking these drugs. Patients gain confidence, we gain confidence. And now biosimilars are way ahead of the same product in the innovator drug. And as Dr. Shane pointed out, Eylea biosimilar is bound to come in the next couple of months, latest by 1 year. And that also, as long as the safety is good, it will go and hit the market.
So if you really look at the history of not biosimilars, history of anti-VEGFs, way back in 2005 onwards, when the innovator molecules came inside, I happened to be there in practice. I'm sure Dr. Thomas was there much before, but then I happened to be in practice when all of this came out. All of these innovator molecules had their share of adverse events, especially sterile inflammations. When Lucentis came into the market, I know there was an issue like this. So likewise, other drugs when they came, I'm talking about the so-called innovator molecules. So for that, we have to understand this process of how it is made. So all these, whether it is innovator molecule, whether it is innovator molecule or biosimilar, all of them are made from a biological process. That means there are cell lines creating these products. So when a biological product is made, one cell line may produce a product which is little different from the other cell line. That means an Eylea produced in 2010 could be different from an Eylea produced in 2021. Not literally different, but there will be slight difference. So, and then there could also be a purity issues while it is being developed. So all these issues are common with innovator molecule as well as the biosimilars. But they have gained experience over time and they know how to reiterate or correct it as fast as possible. And that is why we are seeing these agents without producing any sterile inflammations now. I think these people came into the market lately, so they found a problem which is quite natural to have with any biological cell lines. So when you know this, then we know that it is not because of the biosimilar that this sterile endophthalmitis is happening. It is because of the process of development, which is more or less same for both the innovator molecule as well as the biosimilar.
Okay, so my next question is to Dr. Shane Matthew. Most of these biosimilars are now approved for AMD, in neovascular AMD, and how far it is useful in other conditions also, diabetic macular edema and vein occlusions. And I think Rasamab is approved for both vein occlusions and neovascular AMD, but the other drugs which we have now, the studies are mostly for neovascular AMD. The approval is mainly for neovascular AMD. So how far do you think they are effective in this?
Yeah, so that's a nice question. First, let me answer from my practice point of view. I've been using Rasamab since 2015. I started using it when it was launched to the market itself. I started using it. Probably by now, I would have used around more than 3,500 vials of Rasamab. So I do not have a study to back you and tell you what it is, but I clinically feel that they are effective across indications of, whether it be AMD or macular edema or an RVO case. So it's very difficult to conduct a study on finding the equivalency of efficacy between all the diseases, because every disease is a different etiology altogether, and their response rate is different. But interestingly, there is a CSUS study from Razumab where they compared the efficacy between diabetic macular edema, RVOs, as well as AMD. So they looked into the central foveal reduction in the central foveal thickness as the criteria in judging the efficacy across different etiologies. And the central they concluded by in a 3 months time, they concluded that the central thickness reduction was comparable between all the three groups, namely by 40%, 30%, and 29%. So I would say even studies are also backing my my knowledge, telling me that the response of this biosimilar to all these diseases are quite good and probably comparable.
Very rightly said. And we were using Avastin for all these indications without any trial or without any approval. And now we have...
So actually what happened is, the moment it was banned, Avastin was banned, then we, we really got a shock. And then onwards, I have not used Avastin at all. Till today, I have not used Avastin ever since that ban came in. So then I was in the search for something else. What else to use? And that is when Razumab came in, I started using. Because for me, it was very simple. I use Avastin and land up in trouble, and I don't have a regulatory authority to back me up. But I use Razumab and I have a problem. I have a regulatory authority to back me up. And that was the first idea in using this. And also I used to tell my patients, see, this is our problem. And that's why we are not able to use Bevacizumab. And I started using it. Once I started seeing the results of Razumab, I really thought I should be continuing that. And that is what happened in my practice.
Yeah, definitely. We are having very good results with the biosimilars presently. Most of the surgeons would agree on that, I think. So to Thomas sir, I have a question. Yeah, the need for this multiple injections and the cumulative cost being a main threat for the parent drug. So, and we are switching over to the biosimilar. So are we compromising or anything? Are we compromising on the safety? Since thinking that they're cost effective, and is there anything like that? Are we compromising on the safety with the drugs?
Not at all. If one injection is safe, then any injections are safe for that matter. It's not due to multiple injections that the complications could happen. Complications could happen for intravitreal injections. As we discussed in this forum itself, it has happened for the innovator molecules as well as for the biosimilars. But it's not after multiple injections. So we can use multiple doses. Biosimilars, anti-VEGF has to be used in multiple doses. It has to be given again and again for a long period of time. And it's absolutely safe.
Yeah. As previously Shane Matthew sir had told, the immunogenicity of the drug is an important factor, and that could be the reason for some clusters of sterile endophthalmitis that was reported. But the company, they stopped the batches, they corrected it, and I think there are no such issues now. No differences in it.
The only product where the manufacturer has not withdrawn the drug and corrected is about the brolucizumab molecule.
Absolutely.
For the immunogenicity. Every other company, every other product, they have corrected it. While brolucizumab, they have been telling us that it is safe and these are the indications where you should use it with caution, and it is safe. Not that they have corrected the product, but sooner or later that too will get corrected.
Yeah, so if you, to add to what Dr. Thomas said, again, I started using brolucizumab when it came into the market, and the rate of sterile endophthalmitis quoted by them itself was quite huge. Surprisingly, in the initial time, I had a case of sterile inflammation. Of course, I did vitrectomy with the anti-inflammatory agent, and the patient completely recovered from it. But ever since, what I have noticed in my practice is the number of sterile endophthalmitis is reducing even with brolucizumab. So even if though I feel what the company is not telling us that they're doing something about it, but I'm 100% sure they have done something about it now, and they've refined their processes. And right now, the rate of sterile inflammation with that drug is also less. So only thing is, we are taking this biosimilars to a new level, degrading them by saying that they are producing inflammation. Yes, others are also producing inflammations. That is one thing which I wanted to say. And another thing is, we don't have to worry much about biosimilars because there is a global regulation on biosimilar protection. So it is not like initially when biosimilars were produced and one European Union or US FDA had a different approach on sanctioning them. But now there is a global regulation on biosimilars. For example, US FDA has they need an analytical proof of biosimilar. Second, they want an animal study on the toxicity. I repeat, the toxicity on an animal study, and then a brief clinical study. So it's not just a brief clinical study that let the biosimilar come into the market. There are a lot of other things prior to that as well. So I think we can be rest assured, at least in the coming years, there will be much more stringent regulations regarding use of or production of biosimilars.
So I am winding my question with the last question. Kindly share your personal experience with the biosimilars to everyone. Yeah, Thomas, sir, please.
Yeah, I have used a lot of biosimilars ever since I started using it. And as Dr. Shane has said, in our hospital, we have completely stopped the use of Avastin after biosimilars became available and we started using it on a regular basis. So I have a lot of experience on ranibizumab biosimilars, and we have not had a single complication with these injections, while occasionally we used to have it with Avastin. After starting to use biosimilars, we have not had a single complication, not an endophthalmitis, not an IOP rise, not a corneal edema, nothing. So that is my experience, and the efficacy is as good as the innovator product. I don't say that the efficacy of Razumab is comparable to Paginax, but it's certainly comparable to Accentrix.
Yeah, I think I support what Dr. Thomas said. It's quite effective and the complication rate is less. Interestingly, I think I should be telling this in my forum, in this forum though, because a patient had, a mild reaction following innovator molecule injection and used, thinking that I used Razumab the next time, he did not have any reaction. And there was another patient where Razumab had a mild reaction, but the innovator molecule did not have any reaction after, because I have to keep injecting them, isn't it? So again, I would like to re-emphasize this point of explaining more about the immunogenicity. So it is not just about the impurities or cell line process malfunctioning, which is responsible for the sterile inflammation. It is also something known as ADA. So there are antibodies developing to certain anti-VEGF agents. So this is a latest explanation on why the sterile inflammations do happen. So that means from the thought process of problems happening while purifying the process to even an antibody being developed against these anti-VEGF agents. Especially, that is one of the reasons they are strongly pointing out to say that over time, some of the patients who are taking repeated anti-VEGFs started developing inflammation following again the use of anti-VEGF. So there are a lot more we have to learn, or we are learning more and more things regarding the immunogenicity. So please don't blame it only on the biosimilars. Yeah.
So thank you, Shane Matthew sir and Thomas sir. That was huge information from your side. Thank you. Over to you, madam.
Thank you, Dhanya. I do not see any questions on YouTube. So I think we can conclude the discussions now. And I think today's discussion was very informative. And it was actually like biosimilars simplified. We could learn a lot. And it was the experience of all three of you, all four of you actually, which was coming out. I really appreciate it. Thank you very much. Thank you very much. And happy Onam to all of the viewers and to the panelists and the speakers.
Thank you, Sudha, for conducting this in an exemplary way. And also Dhanya, Valsa, and Shane. Happy Onam to all of you.
Thank you.
Happy Onam.
Thanks.
Happy Onam.
Thank you all. Thank you all. Happy Onam.
Thank you.
Thank you.
Thank you.